Hi there, and welcome back from spring break. My name is Andrea Unger, and here is my journal club presentation, Genomic Surveillance Elucidates Ebola Virus Origin and Transmission During the 2014 Outbreak. A quick overview of Ebola virus. It was first identified in 1976, making it a fairly new virus, and there were actually two outbreaks during that year. One in Sudan in Africa, and the second in the Democratic Republic of Congo, what was formerly Zaire. 318 people were infected during the Zaire outbreak, and for that reason, the name Ebola Zaire is typically used to reference the main species of Ebola, although there are 4 other identified species of the virus. There's a 78% average case fatality, and one of the main reasons is due to organ failure. Hemorrhaging as well as severe hypovolemia both cause the organ failure. But in the case of hemorrhaging, the reason that occurs is because of what is known as a cytokine storm. That is, so many cytokines are released into the body by the body's own immune system that the integrity of blood vessels starts to break down, and so hemorrhaging becomes very common. And for that reason, Ebola used to be referred to as Ebola hemorrhagic fever, or EHF, although now it's referred to by the initials EBOV for Ebola virus, and a second abbreviation that I'll be using quite frequently is EVD, or Ebola virus disease. And speaking of Ebola virus disease, here are some of the symptoms. Mainly what I want to go over is those that cause a lot of fluid loss. So diarrhea, vomiting, and then hemorrhaging, as previously noted, are going to be the main symptoms, and also important for spreading the virus, because people who have EVD are typically not able to be independent. So they're going to be cared for by people, and any contact with their bodily fluids and the mucous membranes of someone who is uninfected is going to spread the disease. So it's a pretty rapid and difficult to control type situation. So in terms of the 2014 epidemic, that began actually in 2013, in the end of the year in December. But over the course of the next 2 years, over 11,000 people died, 30,000 were infected, and most of those were in Guinea, Sierra Leone, and Liberia, although people in the US, the UK, Italy, Nigeria, Mali, Senegal, and Spain were also infected. And one of the primary reasons that it grew so rapidly is because it first appeared in Guinea in an urban center, rather than, as it had been in the past, other areas in Africa that were a little bit more remote. And so there was a doubling time of less than every 35 days. So a pretty exponential growth. Okay, so the study that I looked at really aimed at addressing 2 questions. How did the 2014 outbreak relate to previous outbreaks? Remember that they had been going on since 1976. And then what caused the 2014 outbreak to spread? Was it because of human-to-human contact, or was it because humans were routinely getting it from whatever the reservoir was? So here's the outline of how they did this study. This group of researchers were stationed at Sierra Leone's Kenema Government Hospital, KGH, and there they did PCR-based diagnostics. So Guinea had started to see this outbreak occur, and they were using PCR to start diagnosing people. And up until about May, everyone was coming back negative. The first case in Sierra Leone was from a woman who had attended a burial of an EBOV patient in Guinea. And so she had come back to Sierra Leone, and so she was patient 0 for that country. And then the researchers moved on to the US, where they took their samples with them. They began with 15 EVD samples from 12 different patients, and using these samples, they were able to identify Nextera and Illumina sequencing as their primary ways of being able to sequence the genomes of these viruses. They found that those were the most reliable. Once they had figured that out, they went back and got 84 more EVD samples from 66 patients that hadn't yet been sampled. And at the time, that was, close to 3/4 of all of the EVD patients during the 2-month window in which they were doing their study. So they took these results, 99 results in total, and added them to 3 other samples that they got from the Guinea EVD patients, as well as 17 other sequences from prior outbreaks before the 2014. So in total, they had, 99 plus another 20, 119 samples that they were comparing. Okay, and so here are their results. Up along the top, you can see the genome of the Ebola virus, and there are 7 different genes that they were looking at. And then what they did is they compared them all together and found any areas that there were single nucleotide polymorphisms. They compared those, and you can see that there's either synonymous mutations, those are in green, non-synonymous mutations in pink, or intergenic mutations in gray. As a reminder here for you, the synonymous mutations were any mutations that coded for the same amino acid, so no change in function, whereas non-synonymous are point mutations that could have resulted in a different amino acid and potentially a different function. And notably, there are 3 clear lines at which several mutations occur and are maintained throughout the remainder of the samples. They represent 3 unique haplotypes, and in figure B, you can see that the SL3 haplotype is introduced in early June of 2014, and then quickly becomes the common haplotype among those sampled. So there's some evidence to suggest that that had evolved some kind of adaptation that made it more prolific within the population. So using the data that they had collected along with other sequenced data, Geyer and colleagues assembled a phylogenetic tree using beast dating software. As a quick aside, don't just Google beast dating. You'll get some weird stuff. Anyway, each branch represents a zoonotic event where a human contracted the virus from a natural reservoir. Importantly, the team determined that each outbreak was the result of a single zoonotic event, and then the outbreaks spread from human to human contact. So that's represented by each of the triangles that stem from each branch. And specifically, the team determined that the EBOV that moved through Sierra Leone was the result of 2 separate incidents of human spread into the country. The first grouping of 12 had all attended the funeral of the infected patient in Guinea that I had spoken about earlier, and then a second wave came from a separate individual. So the 2 sets diverged sometime around February of 2014, and both had entered Sierra Leone in or around May of that year. And so here are some final thoughts. To begin with, the team found that the mutation rates roughly doubled during the outbreaks as compared to the time in between outbreaks, so twice as fast during outbreaks. And that likely means that within the natural reservoir, there's some genetic equilibrium and that there's not much evolutionary pressure, but once it enters the human population, there was a lot more of an evolutionary pressure, and therefore a lot more evolution. And they published their results rather early on. They didn't really get a chance to distill the information as much as I think they may have wanted to, because they found that sharing the information was going to be really key to being able to try to figure out where the virus had come from in terms of reservoir and how it had spread. So keeping in mind that they actually published this mid-2014, and the epidemic didn't resolve until late 2016, and it's still not even known exactly what the natural reservoir was for this virus. So being able to share that information quickly was important. And then finally, a quick in memoriam, there were 5 co-authors of the paper who had passed away from EVD during the course of the publication and the research, and I just thought it was important to pay some tribute to them, because clearly this is not easy work, it is not safe work, and it highlights the deadliness of Ebola virus as well as the importance to continuing on with research. And finally, here are the resources that I used in addition to the paper. So if you've got any questions, feel free to post them on Blackboard, and I'll get back to you as quickly as I can. Thanks.